utilize a sensory stimulus. So if we're talking about the knee-jerk reflex, then that stimulus is tapping the patellar tendon below the kneecap. That stretches the quad muscles, which fires a sensory receptor. It goes to the spinal cord. That would be the sensory part here, okay? Goes to the spinal cord, interacts with the neurons going to the muscle, goes out to the thigh, the muscle contracts, and you kick your leg out, okay? So that's the motor reflex for that aspect, using skeletal muscle and a sensory stimuli to cause it to be activated. So is breathing a visceral response? Let's move to that. A visceral reflex would be shining a light in your eye. And the sensory leg of that would be the optic nerve, which is stimulated by the photons going through all the photoreceptors, all right? And then the pupil constricting, and that's smooth muscle. So that would be a visceral reflex, because we're talking about smooth muscle. Smelling food and having saliva produced is a visceral reflex, okay? The sensory being the smell, and the glandular activity producing the saliva being the response. So it's a visceral response. So is the breathing activity that we see here a skeletal motor reflex or a visceral motor or glandular reflex? Pardon? Skeletal, exactly right. Because the muscles that are responding for breathing are the diaphragm and the external intercostals. Okay? And yet, it's a reflex. And you can say, I can bend my knee, I can extend my knee if I want to, or if I had my spinal cord cut and someone tapped my patellar tendon, it would still jerk out, even though I wasn't consciously doing it. So this reflex will work. I can take a deep breath, I can take a shallow breath, I can hold my breath, hence the voluntary aspect of it, but I can be asleep and not think about breathing. And because of the stimuli, still respond by contraction of those muscles. Okay? So it's kind of that triangular effect that we're looking at. So this is going to be based, the stimuli are based on receptors that are present in the blood for those three major chemicals, all right? So if we look on, you have this particular diagram, and this is where I changed it around. In fact, I'm going to go do that right now and come back to this here. So where we talk about being on the fly. All right, so this is the diagram that you have in your lecture notes. And these are the structures that we look at in the control of breathing. So if we look at the pattern, that triangle that I had on the board, on the screen, we had the control center, central control center. We have the sensors and the effectors or muscle components, all right? So looking on this diagram, the control center is right here in the medulla, okay? The medulla oblongata, and in the pons. So we would have the pons, which is the secondary control center, and the medulla containing, if you recall from Bio 430, the respiratory center. Okay? Influencing that central control center are, so here would be the control center right here. Influencing that control center would be the tissues above the brainstem. So the frontal lobe, emotions, you can voluntarily change how often you take a breath. All right, pain can make you breathe more rapidly. So emotional system, the hypothalamus can send descending information. So that's what we see here. So these components are coming down to the center in the pons for, let's say you're too hot. You breathe more rapidly, okay, to cool off. Get rid of some of the heat that's in the air that you're breathing out. And so then we have voluntary control as well with the frontal lobe, okay? So what about the sensory information coming in? Obviously, if it's pain, if it's, temperature, that would be descending down from the upper portion of the brain. So pain, temperature control would be coming down from the upper portion of the brain. But we have our visceral responses as well, coming from peripheral chemoreceptors. And coming from central chemoreceptors. And we'll look at these in a little bit more detail. Chemoreceptor refers to what type of information this is sensitive to. Chemicals, all right? And the three basic chemicals are carbon dioxide, hydrogen ions, and oxygen level for the peripheral chemoreceptors. And central chemoreceptors, I'm going to put carbon dioxide in parentheses, and hydrogen ions. So as these levels change, they can modify the rate at which we breathe. All right? So I'm going to leave that aside, and we're going to look at just the typical respiration pattern, and then we'll come back and look at a little bit more detail how these particular chemicals can modify the rate. So we have a general rate at which we breathe, and then we can modify that. Does that sound familiar? Familiar to what? We have a general rate at which our heart beats. Exactly right, our heart beats, and then we can modify that, okay? So keep that in mind. But before we leave this diagram, the sensory information coming into the control center then is going to send the outward leg of our reflex to our muscles, okay? And this outward leg is going to descend down the spinal cord and synapse with neurons that exit the spinal cord. And where are these neurons going to go? What neurons would they be, by the way? So this is going to control the respiratory rate and the depth of breathing. So C3, C4, C5. What nerve would this be? So this is coming down to the level of C3, C4, C5. We know what nerve controlling respiration exits from that level of the spinal cord? Goes to the diaphragm. Phrenic. Okay. So that's the phrenic nerve going to the diaphragm. And these would be T2 through T12, the intercostal nerves. Going to the external and internal intercostal muscles. Okay, so that's our overall pattern. Something in the medulla, affected by the pons, is going to trigger a respiratory, an electrical current at a particular rate that will travel. Its rate can be modified by changes in carbon dioxide or hydrogen or oxygen levels. Its rate can be modified by stress or emotions or body temperature. And then the final decision on that rate will travel down the spinal cord to our voluntary motor neurons of the phrenic nerve and the intercostal nerve. Okay? That's kind of the whole big picture. So now, going back to the similarity that you recognize with the heart, what did we have in the heart that didn't require neurons? The SA node, the pacemaker, the autorhythmic cells that depolarize on their own at a apparently constant rate, SA node about 100 times a minute, and we use the sympathetic and parasympathetic nerve fibers to modify that rate, okay, based on oxygen needs and blood pressure requirements. So we have a similar, less understood regulation of our normal breathing rate. So, a lot of words, and I'll put this up on D2, I'll kind of modify it a bit. Number 1 up there says the pre-Botzinger complex. And that pre-Botzinger complex is, let's go back and forth between the 2 images. I actually have this diagram behind the board. But if we go back to this diagram here, within the medulla, we have our respiratory center formed of a dorsal respiratory group. Let me just make this bigger here. And a ventral respiratory group. Okay? These are cell bodies of neurons that are found in the medulla, and that's what forms our respiratory center. And then up in the pons, we have another area. Back in I think the first paper was actually printed in 1991, an area, a group of cells that were found to be up here, kind of at the top of the ventral respiratory group, was a cluster of neurons known as the pre-Botzinger complex. Love that name. And this group of neurons was discovered to depolarize on their own. Okay? So they are now thought to be the source of the generation of the respiratory breathing cycle. And then they are modified by these other components. But they are the ones that, and why they actually do that, why they automatically depolarize, we haven't figured out yet. Okay? But they are thought to be like the SA node, just to depolarize on their own, and then recover, and then depolarize again, and recover, and depolarize again. And that sets up the generation of our respiratory pattern, okay, which we then modify. So back to this slide. So the pre-Botzinger complex arrhythmic pattern of breathing arises from a network of spontaneously discharging neurons. And that's what I drew in that purple circle up there. All right? In response to that, then, the dorsal respiratory group, which is formed only of inspiratory neurons, appears to function in our normal everyday breathing. The 2 seconds in, the 3 seconds out. 2 seconds in, 3 seconds out. The ventral respiratory group has both inspiratory neurons and expiratory neurons. This is continually changing, all right? In preparing for this lecture, I found almost contradictory information out there about the role of each of these. Your current textbook says the dorsal respiratory group is in charge of starting breathing. I went to a meeting where I got just a preview of a chapter from a new textbook. It said the ventral respiratory group was in charge of breathing. It didn't give any citations. And going back and reading more about the pre-Botzinger complex, the reason I think they say that is that the pre-Botzinger complex is now thought to be part of the ventral respiratory group. Okay? But it will trigger apparently it triggers the dorsal respiratory group to fire. So the rest of the ventral respiratory group is still thought to be functional in forced expiration or accessory inspiration. So So if you need to breathe deeper or if you need to force air out, we use the ventral respiratory group. Okay? And we'll look at each of these in a little bit more detail. So that's the medulla. So we start with the pre-Boxinger complex with just our pattern generator. Breathing in, breathing out, breathing in, breathing out. Okay? That pattern turns on the dorsal respiratory group, which would then send neurons down to the phrenic nerve and intercostal nerves to trigger you to take in a breath. Then they stop and you breathe out. Okay? And then they fire again because the pre-Boxinger complex has depolarized. And that's kind of our normal pattern of breathing. And then the ventral respiratory group would come in to forceful, take a deeper breath if you have to, if you're high altitude, or blow out harder if you're blowing up a balloon. You would activate those ventral respiratory group neurons. Okay. Finally, we have neurons in the pons that tend to do what the cerebellum does for our motor movement, kind of smooth out movements. So we'll see what happens to our breathing rhythm and smoothness when the pons is not available. Okay, so we'll look at that as well. So it either it limits how much you can breathe in, okay, or it affects your total inhalation. And then we'll come back and look at the very last one down here, those of you in the back. It says ventilation is subject to modulation by various chemical factors, carbon dioxide, hydrogen, oxygen. Okay, so we'll come back and look at that aspect. All right, so generation of a rhythmic ventilation. Starting inspiration. So here's where we modify. These various factors will modify that. But the starting inspiration, the generation from the pre-Boxinger complex, and if we don't have anything abnormal, we modify it with this. The dorsal respiratory group fires, what we just went through. Okay, the phrenic nerve is activated, the intercostal nerves are activated, the diaphragm is lower, the ribs are lifted up. Intraalveolar volume is increased, atmospheric pressure is higher, pushes air into the lungs. Okay? All of that occurring as a result of the dorsal respiratory group firing. This increases, and you breathe more and more and more deeply because of activity known as the ramp effect. It's basically what you learned in Bio 430 as recruitment. And then we stop inspiration, and we stop breathing for about 3 seconds as all of those muscles relax. All right, so that's kind of the overall view of that respiratory pattern. So we're going to go over this multiple times. Okay. So the dorsal respiratory group, here's the same diagram again. Here is the medullary respiratory center. Dorsal respiratory group is a little higher and dorsal, closer to the cerebellum. Ventral respiratory group is a little lower and more anterior, closer to the ventral surface. Okay? And then our Boxinger complex would be located kind of in between the two at the top surface of that group. So the ramp effect looks like this in its most simple effect. So the pre-Boxinger complex starts to depolarize, right? And our dorsal respiratory group neurons are activated, and then more are activated, and then more are activated. So how does this translate to a deeper inhalation? More recruitment. We start out with a lower amount of current. We pick up the respiratory muscle cells that have the lowest threshold. And then as we generate a higher current, we pick up those muscle cells that have a higher threshold. And we have a greater and greater force of contraction, lowering the diaphragm, lifting the external intercostals, lifting the ribs. Okay? And then it ceases, and that's the 2 second, and then we have the 3 second gap. And then it starts up again. All right? So this would be the pattern generated by the pre-Boxinger complex, activating the dorsal respiratory neurons, known as the ramp effect. And so you can see it here, breathing in for 2 seconds, passive expiration for 3 seconds, breathing in for 2 seconds. That's a pattern generated, and then activating the dorsal respiratory group, which then activates our respiratory muscles. Okay? So we can also see that on this cycle. So this is quiet breathing, no stress, no pain, no cold, sleep, vegetating on the couch, no exams to study for. And the pre-oxygen complex fires, the dorsal respiratory group fires, the ramp effect occurs, inspiratory muscles contract, inspiration occurs, and the dorsal respiratory group is inhibited, typically by the pons group. They relax. Expiration occurs for 3 seconds, and by that time we're ready to start again. Okay? So this is the basic pattern of respiration without consideration of change in CO2 levels, or exercise, or low oxygen. Okay? So no other changes to the chemicals, just everything's normal, no other modifications that the body needs to adjust to. Any questions on that? Okay?
Yeah, can you point out the ramp effect on that diagram again?
The ramp effect would be taking place here.
Okay.
So that first spike of a current would start the first muscles contracting, and as higher and higher spikes occur, they just do it more deeply. So as you're breathing in, the reason you're getting more air is the diaphragm is lowering not just a tiny bit, but there's a ramp effect means we recruit more muscles, therefore there's a stronger contraction, the diaphragm is lowered more. Okay? All right, so the dorsal respiratory group would be involved with the onset of passive expiration by which of the following? Expiratory neurons in the dorsal group would be activated, or inspiratory neurons in the dorsal group would no longer fire. Yes, because remember, the dorsal respiratory group is formed of only inspiratory neurons. So we exhale by stopping their activity. All right, so we stop that activity and it's a passive expiration. Okay, the dorsal respiratory group receives input from the peripheral chemoreceptors. Yep, we saw that right here. Okay, we'll talk about where those peripheral chemoreceptors are. It receives direct input from the limbic system. Well, that's on your picture on page 68. Does it receive direct input from the limbic system? No. That goes to the pons first, and then from the pons to the dorsal respiratory group. Okay, so that one would be true. Sends efferent neurons to the pons. Nope. Is responsible for forced inspiration or exhalation. Nope, that's the VRG. Okay, so just this one would be correct. It's actually both peripheral and central. All right, so let's look at the ventral respiratory group then. It also has inspiratory neurons, but this group also has expiratory neurons. Okay, and so we can increase, because of the presence of the inspiratory neurons, we can increase the normal inhalation, that's the forced inspiration I talked about with the ventral respiratory group. And because of the expiratory neurons, now those are going to be going to the abdominal muscles and to the internal intercostal muscles for forced expiration, blowing up a balloon, not just the passive relaxation. Okay? So a cycling image of that would be, I didn't want that slide there yet, would be here. So both the dorsal respiratory group and the VRG are active. So we have inspiration. We actually have forced inspiration, okay? And then the expiratory group of the ventral respiratory group is active, and we have forced exhalation. So we can do both, or we can do just the forced expiration. So what you were doing when you were using the spirometer last week, you were using your VRG groups. Okay, to try to take it as deep a breath as possible and then force it through that spirometer. So you were using both your dorsal respiratory and your ventral respiratory groups, okay? So the pontine respiratory group and the apneutic center they're required for you to be able to breathe. Would you still have, if we cut off any connection between neurons of the medulla and the pons, or the frontal lobe for that matter, would you still breathe? All you had was the medulla. Correct. You would still breathe. It would be a gasping breath, okay? Like that. But you would still have sensitivity to carbon dioxide and hydrogen ions, and you would still have the lower motor neuron connection to the diaphragm and the intercostal muscles. But without the pons, okay, we don't have this fine tuning. What part of the brain is responsible for fine tuning of our motor activity, our voluntary motor activity? Not the breathing voluntary, but if I'm going to reach out and pull this over. Cerebellum. Okay, so in essence, the pons respiratory centers here are acting like the cerebellum to smooth out the respiratory pattern, okay, and to fine tune it. So it can inhibit the DRG. This is when you stop. This is you breathe in, and then this fires to inhibit the DRG and allow that 3 seconds of breathing out. Okay. And then the next portion also does fine tuning. This keeps you from just stopping breathing all completely. Right. So again, there's a lot of gaps in our knowledge on how this all works together. And this information comes from working with monkeys and with cats primarily, deliberately damaging their brainstem and seeing what happens to the pattern of respiration. It also comes from humans who have had brain trauma, okay, aneurysms or other areas, and recognizing, looking at their breathing pattern and looking at autopsy reports. So, well, okay, what part of the brainstem was damaged? We haven't quite gotten to the point yet where we deliberately do trauma to people. Okay, but those are the studies that have shown this kind of information. So here's a diagram. Don't worry about all the stuff that's in here. Okay, that's anatomy and nuclei that we haven't dealt with. But here's medulla. Don't have this in your lecture notes. So here's the medulla. Here's the pons. Here's the midbrain. Okay. So if we were to cut below the medulla, say at C1, straight line, no breathe, not a straight line ECG, but no respiration, all right? We know that from the fact that the phrenic nerve starts at C2, C3, well, C3, C4, C5. So damaging, severing the spinal cord above that means there's no connection between any control, voluntary or reflex control, to our muscles of breathing. All right, so you cut below the medulla, no breathing whatsoever. Now, you cut between the midbrain and the pons. Remember the question I asked? Can I answer correctly? You will breathe. All right, if we have only the dorsal or respiratory, ventral respiratory groups, and here you have this irregular gasping. So you will breathe, okay? Here's the fine control that the pons provides. If we don't have the pons breathing, but in and out, but irregular, we add the pons, and we start to see, depending on what level of the pons, we start to see a smoothing out, a fine tuning. And that's what I wanted you to get from this diagram, okay? Regardless of where the nuclei are that are being cut, what is happening, okay? Why? So, no breathing here because we've severed any connection to the spinal nerves. Breathing does occur here because we still have our medullary respiratory center, but not smoothed out. Here, breathing is smoothed out. It may not be able to respond to frontal lobe changes or temperature components because the limbic system connection would be lost, but you would still have a more regular pattern of breathing. And we see this in infants that are born with loss of brain cortex. or the merencephaly, where they have just a brainstem and different parts of the brainstem may or may not be functional. Okay, so let's go back up to the top now and look at those factors which can change this basic rhythm. All right, because those factors that we just looked at were assuming that none of those basic rhythms were affected. So these are the chemicals that the chemoreceptors are sensitive to. Carbon dioxide, hydrogen ions for arterial pH, and oxygen. Notice the word prior to each of these chemicals is arterial. Okay. So the receptors, the chemical receptors, are in arteries, not in veins. And before we move into that, let me illustrate. Here is an alveolus. All right, here's arterial blood, what's going into the arteries. And so we see equilibration, all right? So in a normal situation, we're going to have normal levels of CO2, blood, the oxygen dissolved in the blood. And that arterial PO2 is, remember, approximately 104 millimeters of mercury. Okay? Finally, something you feel comfortable with. And our normal PCO2, as we're leaving the lungs, is going to be what? Anybody remember? 40. Okay? And normal pH is, we'll round it up, 7.4. It'll range between 7.35 and 7.45. So now that blood is carried to the tissues, and at the tissues, oxygen is going to be used up and carbon dioxide is going to be added. All right? So the blood returning from the tissues is going to have a partial pressure of oxygen of 40 and a partial pressure of carbon dioxide of 45. And a pH, we'll say it's slightly acidic because of the carbon dioxide that's in there, okay? So, if we were dependent on these numbers, all right, slightly elevated PCO2, we would take a deeper breath, all right, would be changing our respiratory activity. But that's the effect of just utilizing the tissues. And as it passes into the lungs, those changes are going to occur normally because of lung function, right? We're going to have carbon dioxide exchange, and CO2 is going to leave the blood, and oxygen is going to diffuse into the blood. And as we lose CO2, our hydrogen ion levels will fall and our pH will rise. That's just going to happen with a normal lung function. Now, let's change that normal lung function, okay? And let's say that this individual has pneumonia and they've got an exudate in their lungs. And we've thickened the diffusion distance, all right? So now, how will that change the blood that is leaving the lungs? Would you expect to have a normal PO2? Because of our greater diffusion distance, that value is going to be lower, all right? So, depending on how bad the pneumonia is, we'll just put lower. PCO2 depends. Carbon dioxide diffuses much better across the membrane. So we're going to see changes in our oxygen much more quickly than we'll see increases in our PCO2 because it has such a high solubility. Remember, it's 20 times more soluble. But let's say, for argument's sake, we've got pretty bad pneumonia. So our PCO2 is going to rise because it has to diffuse out across a longer distance. And that better solubility of CO2 isn't helping us much because it has to diffuse through that increased distance of fluid in our pneumonia. And so because of the higher PCO2, we're going to see a decrease, a drop in the pH due to an increase in the hydrogen ions from our carbonic acid. Okay, everybody with me on that? You visualize the problem that we've had. We can't get rid of the CO2, and we can't get enough oxygen. And because we can't get rid of the CO2, we have more carbonic acid, and therefore more hydrogen ions. Okay? Yes?
On the purple pH, is it negative 7.35?
No, that's supposed to be equals. Just a connecting line. All right. So now, notice our arterial pH, our arterial blood gases are no longer normal. So our chemoreceptors are sensitive to arterial values rather than venous values because those change all over the place. And there's not a problem as long as the lung tissue is working well.
Should the pH of that be lower than 7.35?
Yes, that's why I put the arrow down. I just don't know how much that, okay, I'm saying that the pH has dropped. Okay? So that's why our chemoreceptors by which we use to modify our regular respiratory pattern are sensitive to arterial levels. Because we would expect those other levels to change with metabolic activity very wildly. Okay? The key is the arterial. Those are abnormal. That's when we need to see a change. So our chemoreceptors are sensitive to these three values. We have two locations for these chemoreceptors, all right? We have central chemoreceptors, and this is back to the pattern of what you learned for central nervous system and peripheral nervous system, okay? So the central chemoreceptors are in the medulla, and they are found, if this is, rather than, again, I have this drawn behind, but here's the cerebellum, here's the medulla. This is the fourth ventricle. What would we find in that fourth ventricle? You guys remember your CSF, okay? Here's the pons.
That was a trick question. I was like, something else.
And here's the medulla again. So these central chemoreceptors are here in the dorsal surface of the medulla. Just put a legend up here, central chemoreceptors. And up there on the slide, it doesn't have the word oxygen. It's carbon dioxide and hydrogen ions in the CSF. Notice it didn't say arterial hydrogen ions. Ever heard of the blood-brain barrier? Okay, so capillaries in the medulla, and I don't have a red pen, but let's say that this is the capillary in the medulla. We'll make it take up the entire medulla here. Hydrogen ions in the capillary cannot cross the blood-brain barrier. They can't leave. So you can have really high hydrogen ions in your arterial blood, and the central chemoreceptors are not sensitive to it at all. All right? So we have what is an indirect pattern. We also have carbon dioxide dissolved in the blood. Carbon dioxide is a gas. It can cross that blood-brain barrier. And guess where it crosses to? into the CSF. All right, so here's a diagram of that. So here's the capillary. Here's the hydrogen ion that can't get out because of the blood-brain barrier. Here is the CSF. And here's the carbon dioxide gas that can diffuse out of the capillary. All right. Notice, what does CA stand for? Anybody remember from that day before spring break? Carbonic anhydrase. That's exactly right, Karen. In our normal blood, where do we find CA? Carbonic anhydrase. And we talked about it as a mechanism in the transport of carbon dioxide. Where was carbonic anhydrase? Was it in the plasma? Inside the red blood cell. Okay, and that's where we formed the vast majority of our carbonic anhydrase. It's one of the most rapid chemical reactions in the body. Here, it's secreted by the cells lining the ventricles, and it's actually found as a component of CSF. So, CO2 diffuses into the CSF. Within the CSF, it makes carbonic acid. Carbonic acid dissociates some of it into free hydrogen ions and our bicarbonate ion. Here is our receptor for hydrogen. This is the chemoreceptor for hydrogen. All right, so there is no chemoreceptor for the hydrogen in the blood, but for the hydrogen in the CSF. As CO2 builds up in the arterial blood, then more carbonic acid is made in the CSF, and more hydrogen binds to the chemoreceptors along the ventricle, and those send a signal to our ventral, our Botzinger complex, but primarily to the dorsal respiratory group, and it fires more rapidly. Okay? So that's why in this list, arterial PCO2 you can put in parentheses, okay? Because it's not the direct concentration of arterial PCO2, but the hydrogen ions it forms within the cerebrospinal fluid. All right, so let's go back to that first question I put up on the board. Which of the following chemicals is the primary CNS factor in stimulating a change in respiratory rate or death? Well, it's still a tricky question. If I phrased it, which of the following factors activates a chemoreceptor, then it's going to be D, CSF hydrogen ions, okay? If I say which factor in arterial blood is most important in changing respiratory rate in the central nervous system, it can't be this because that's not in arterial blood. It can't be this because hydrogen ions can't cross the blood-brain barrier. So it would be this. Does that make sense? Okay, anybody want me to rephrase that? Okay, so it's critical on how the question is phrased, because obviously the hydrogen ions in the CSF are directly relevant or dependent on the amount of CO2 in the blood. Okay? So you can't just say, well, which is the most important, CO2 or hydrogen? Well, where? Okay. Elevated CO2 in arterial blood will mean elevated hydrogen ions in the CSF, and that is what actually binds to the chemoreceptor. Okay? So there are no oxygen chemoreceptors in the central nervous system. We find those just in the peripheral nervous system. Okay? So let's look at that, and then we'll compare why these two factors vary. So peripheral chemoreceptors are sensitive to all three. Peripheral chemoreceptors are found, and I've got one location over here on the side. They are found within the lining of arteries. So we don't have to worry about a blood-brain barrier. And we're not in the central nervous system, so we don't have blood-brain barriers anyway. But over here, this vessel is the common carotid artery. It's dividing to form the external carotid artery and the internal carotid artery. Okay? So right at that bifurcation is a little bump in the lining of the blood vessel known as a carotid body. That dilation is called the carotid sinus. So kind of the sinus is the dilation, the bump is the carotid body. Another location where we have this is in this vessel. The aorta. So in the aortic arch, we have the aortic body within the aortic sinus of the aortic arch. Okay? Arterial blood, right? Carotid arteries, arterial blood, aorta, arterial blood. So reinforcing that aspect again. The sensory neuron that is activated by this in the aortic is a component of the vagus nerve. The sensory neuron that's activated by this in the internal carotid artery is the, I want to write the whole thing out, the glossopharyngeal nerve. Okay? And they will carry this information back to the dorsal respiratory group. So I'll show that to you on the diagram. So the most important one here is the pH and the CO2. Why? Because the central nervous system doesn't have those. So we get sensitivity to hydrogen ions in arterial blood via the peripheral receptors. And we get sensitivity to oxygen in arterial blood in the peripheral nervous receptors, Okay? And I'll give you some examples about that. So here's an image of that glomus cell. Okay, right there at the bifurcation. And here's how it works. This is just oxygen, okay? The hydrogen ions and the carbon dioxide ones I haven't drawn. So arterial blood is flowing up to the brain and to the face and is going to contain relative amounts of oxygen. If the oxygen is normal or high, these potassium channels stay open and potassium slowly leaks out. Okay, we have potassium pumps that pump it back in again, but we have potassium slowly leaking out. However, let's say you're climbing Mount Everest and you're at 18,000 feet and you're trying to do it without oxygen. CO2 is going to drop, right? You're going to have less oxygen in your arterial blood. And this happens when oxygen falls below 60 millimeters of mercury in pressure, okay? With low oxygen, the potassium channels close, and if potassium can't leave, what's going to happen to the number of positive charges in this cell? They're going to go up, okay? Hence, the cell depolarizes. Now, I diverge for just a moment. We look at a typical neuron with the synaptic gluton. As the electrical current travels down that neuron, it reaches calcium channels. And in response to that current, those calcium channels open. The calcium ions bind to the synaptic vesicles and allow them to release the neurotransmitter. Almost identical things that we're talking about just